Good morning, everyone, and very welcome to this edition of the Meet iToBoS series. This morning, we are very happy to welcome our colleague Claire, who's coming from far Australia, but right now not from quite so far because she's luckily in Europe, so the time zones are a little bit less of a challenge. We wanted to give you like 2 lines of introduction what this project is about. So iToBoS stands for Intelligent Total Body Scanner and is for the early detection of melanoma. So we're in melanoma, so I took that for a given. And it is a project that is funded under the Horizon 2020 program. And MPNE, so the Melanoma Patient Network Europe, is one of the project partners. And we are responsible for patient engagement, among other things. And one of the feedback we got from our community was that people were very interested to know what is going on inside the project. And we, of course, had the opportunity to talk to our colleagues and find that out. So we're now playing or testing different type of formats to give everyone the opportunity to find out more what's going on, have a discussion with our fantastic colleagues. iToBoS is a particular project because it is very broad, so it has very technical aspects. It includes building a prototype, so that's engineering, there are lenses, there is artificial intelligence, but there's also a clinical trial in there. So that makes it a really interesting group of people because it's so diverse and everyone working together to hopefully achieve one final product. So today, Claire will tell us exactly of what I just mentioned, namely the clinical trial that is part of iToBoS. Claire, it's an absolute pleasure to have you here. I think it would be great if you gave us a little bit about who you are and your background, your role in the project, and then walk us through the clinical trial. Looking forward to this.
Absolutely. Okay, I'll just bring up my slides. Oh, how do I? Oh, there we go. So firstly, just a bit of a self-introduction. So my name's Clare Primiero. I'm from Australia. And I predominantly work for University of Queensland, which is leading work package 10 for the iToBoS project. But I'm currently lucky enough to spend a year or 2 in Barcelona in Spain, where I'm also working with one of the iToBoS partners, which is the Barcelona Hospital Clinic in Barcelona as well. So as for my background on how I ended up here, I completed a Bachelor of Science with University of Queensland in 2009 in biomedical science. And I spent a couple of years working in a research group at UQ, working on microneedle technology for delivering vaccines through the skin rather than the needle and syringe. And then from there, I was persuaded to join the Dermatology Research Centre, which is also part of University of Queensland. And I've been there ever since. So, but over the last decade, my position has really kind of changed from that real lab-based immunology type studies to more of a clinical research role and being involved in melanoma research, particularly focusing on early detection and stratifying risk to, make sure those that need to be seen most are actually being seen. So during this time, I've also completed a master's in clinical trial research with the University of Sydney. And also I've recently completed a PhD just last year. And my PhD topic was on providing genetic testing for familial melanoma. And part of that was piloting a training program to be able to train clinicians to be able to provide testing. And I designed a clinical trial so we could test this against the standard care, which is genetic testing received by a genetic counsellor. And the idea is that we think this is something that could be mainstreamed into dermatology clinics to make it more accessible. So it was kind of a first step just to pilot this program. And probably my greatest achievement over the last decade would be these 3 as well. So that's really my background. Here, I feel quite fortunate that I'm able to be working on iToBoS and come to Europe for a year and work with the partners here face to face. It's very cool. So I know everyone's probably familiar with iToBoS, but just to summarize, the main objective of the iToBoS project is to develop an innovative diagnostic platform for early detection of melanoma. And the two methods of driving this is the total body photography, which differs from existing total body photography because it uses very high-tech liquid lenses that are estimated to produce photo quality similar to dermoscopy. So it would skip that extra step of having to do individual dermoscopy of each lesion. And then also creating the AI tools to create a AI cognitive assistant, which would evaluate risk not just of individual lesions, but in the context of the whole patient. So being able to consider medical background, the total body images, like how many nevi they have. If someone has a lot of nevi, then they are at higher risk, and skin color and as well as sun UV damage as well. And then there's also a genomics component as well, being able to integrate all that information. And so that's what really sets these AI tools apart from what is really emerging at the moment, where most of the AI for skin image analysis is only looking at just a single lesion. It's not in the context of the patient. So that's where it kind of lacks that real world application currently. So I think iToBoS is really addressing that point. So the clinical data acquisition study, that is led by University of Queensland as part of Work Package 12. It's the first clinical study, and the objective is to recruit 500 people to basically develop a training data set that's required for the machine learning to develop these AI tools for risk profiling skin lesions and patients. So from the 500 people that we want to recruit, we're collecting three main sets of data. So the photography, the total body photography, genetics, and then also the medical background information. So in order to collect that information, we've used a purpose-designed survey rather than the option of accessing medical records. So that way we're only collecting the precise information that we need. And this is also based on previous validated surveys on information that we know is associated with risk of melanoma. So the original plan for the first clinical study was that we were going to use the iToBoS scanner to develop, conduct these research studies. However, we all know that there were delays in the development of the iToBoS scanner machine. So we had to use Plan B, which was actually in the original grant application. Plan B was to instead start collecting data using an existing system. So the image here is of the Vectra 3D total body machine. And this is from our clinic in University of Queensland in Australia. We've actually had one for quite a long time. We actually had the very first prototype, which was like a scaffolding with cameras attached to it. So it's come a long way since then. And it already does have some AI components built into it, such as the automatic detection of nevi and being able to provide a count for nevi. And that's what you see there on the right of the screen. And so, as I said, Plan B was to instead, we would use images collected from the Vectra total body machine. And the manufacturers of Vectra is Canfield Scientific, who are now also a research partner in the iToBoS project. So it's been really good to start developing it this way. So how are the images used? And more importantly, how do we do this? How do we train an AI system but still make sure that we're protecting patients' privacy? Obviously, when you're talking about total body images, especially 3D avatars, they're very identifiable and also sensitive, of course, because generally patients are in their underwear for the imaging. So we really had to consider how we can do this and protect privacy. So instead of using the 3D avatar, what we do is we actually go back to the macro photos that are taken from each of the individual cameras within that framework of the Vectra machine, and we use those images instead. And then the first step is de-identifying. So the whole head is just cut off. So we're just not using the face at all. And then there's a second step to anonymize any kind of features such as tattoos or identifying scars as well. And then we cut up the images into smaller tiles like you see on the right of the screen. And these are the small tiles that will be shared from the clinical sites. So before that, these images are only dealt with the clinical sites that are doing the imaging. So only Barcelona will see the total body imaging of the Barcelona patients, and likewise for UQ. And it's only the final smaller tiles that are shared with the other partners and are used for the annotation process that's required to be able to train the AI to analyze these skin images. And this here is just an example of the database that's used by another iToBoS partner called V7. So they provide this database that basically we are able to upload our skin images onto, and then other partners then do the annotation steps using this kind of software, and then from there other partners develop the AI tools. So it's a very multi-step process that there's been a lot of working out on the way how each step goes, but it's exciting to see it all happening. And we were also asking the annotators to put a label to every skin image on the sun damage or UV damage that of a skin. And this is because, likewise, how I was saying, that we want the AI to better replicate a clinical scenario where a dermatologist is also going to look at the rest of the skin and say, oh, well, this person's had a lot of sun damage and they are at higher risk. So we want to be able to develop AI programs to automatically profile this type of risk. So we call it the skin phenotype, one part of it anyway. So there's an option of 3 different sun damage tags that they can add, either the low to none, moderate, or severe. And we provide our annotators with examples to help them label, add this label to the skin images, just shown here. So that's the images covered. Like I mentioned, we've got a purpose-designed questionnaire built on previous validated surveys. So there's 45 questions in total, and it covers 4 different categories, and it takes around 10 minutes to complete as well. So it's not too long. So we covered demographics. And one of the things we try to consider again is privacy. How do we minimize risk on this information being identifying to people? So, we're making sure we're collecting age or year of birth rather than date of birth, and the country of birth and residence, and not, a more specific address. And so it's all kind of just trying to keep it low risk for re-identification. And so, of course, skin cancer history is all very relevant to your future skin cancer risk. So we collect that information from personal and family history, as well as past and present sun health behavior, how often you get your skin checked, your history of sunburns or sunbed use, and then phenotype information. So we collect self-reported data on your natural hair, eye, skin color, and skin type, whether you easily burn or tan, as well as like a judgment on your amount of nevi. So we count that into 3 different categories. It's like none to few, some, few, or many. And we give them a picture diagram on what they think best suits them. But at the same time, we're also trying to train the algorithms to automatically extract a lot of that data as well from the images as well. Because we know that self-reported data cannot be too reliable in some points. So we're working on both of those simultaneously. So genetic information, I mentioned that we're also trying to incorporate genetic risk as well. So for a small portion, probably about 10% of the participants will have like a strong enough personal and family history that indicates more of a genetic link, that it might be familial melanoma, where when you have a mutation in one of these genes, it's associated with a greater than 50% lifetime risk of developing melanoma. And it's also passed on from one generation to the next. So if someone has a mutation, their children would have a 50-50 chance of also then carrying or first-degree relatives would have a 50% chance of carrying the same mutation. So there is a panel of genetic testing of a bit over 10 genes that we know are associated with this that we can test for people that fit this criteria. But for the most part, participants we will be calculating their polygenic risk score. So polygenic risk refers to the cumulative effect of low to medium risk genetic variants. And we're able to look at all those and then add them up. So some of them are associated with decreased risk and others increased risk. And again, in the consideration to protect privacy, DNA is very identifying. So we won't be sharing any kind of DNA sequence past the hospital clinical sites, again, like the images. What we will instead do is calculate the risk score at the hospital sites. And that score will be presented as a number from 0 to 100. And that is the number that will be shared with the other iToBoS partners that are then working on the AI algorithms to risk profile people. So that way we won't be sharing any actual DNA sequences. So just to keep that information confidential. So the clinical data acquisition study that's currently in progress at 2 different sites. When we had to go to plan B and use the Vectra, it meant that the 3rd clinical site in Trieste in Italy wasn't able to contribute because they didn't have a Vectra machine. Originally, all 3 sites were supposed to have the iToBoS scanner. So instead, both UQ and Barcelona, we've increased the amount of participants we've recruited to make up for that. And we've taken 2 slightly different approaches to collect the same data at the 2 sites. At UQ, because we've had the Vectra machine for several years, we already had a lot of this data collected. And so in order to reuse data that's already available and make sure we're using the best of people's time, our participants' time as well, we opted to do a retrospective study. And in what we did is we contacted our past research participants to ask if we could use their previous total body images and their previously connected genetic sample, and then also ask them to complete a prospective questionnaire. And we did this in a new way that we hadn't done. I created an online consent form, and I did that with one of our other iToBoS partners at Trilateral that focus on ethical social considerations in research. And we're quite pleased with the outcome because instead of the paper form, it was more like a PowerPoint presentation. So there were photos, less text, more images. And I think it was quite well received from the feedback that I got. And it was also just very streamlined. So initial contact was via email to our past participants. And that email gave them a short description. And then it was, if you're interested, you can click this link to the participant information online form. And then they were able to go on to consent if they were interested. And then after, they would also get a link for the questionnaire. So we've recruited 245 patients at UQ through this method. And just over 90% finished the questionnaire as well. So some people didn't complete it, but that's all right. Still happy with those results. And we didn't quite get to the 250, we were 5 short, but I would have had to amend the human ethics to include another cohort to be able to get the extra 5. So instead we were like, okay, at Barcelona, we'll just recruit an extra 5 there instead. So in Barcelona, because they haven't had the Vectra quite as long, and also because they are an active hospital clinic and they have patients coming in every day for total body scanning. So it was more convenient for them to do a prospective study and to recruit people as they come in for their standard skin checks. And if they're already having total body photography, then the dermatologist first mentioned to them the study. And if they're interested, then either myself or one of my colleagues will talk to them about, go through they use the paper consent form. I was a little bit surprised that they thought that the online, or even if I used an iPad, they thought, oh no, that's not going to go here. They didn't think that was a good idea for the cohort of patients that they had at the clinic. So, you know, different things work for different sites. So there we're up to about 150 patients so far. And we're hoping to complete this study in Barcelona before the end of the year. And as I said, we're collecting the same data though, even though it's slightly different methods. We're doing the total body photography, and those images are cut up, de-identified and cut up into the smaller skin images. We're collecting saliva samples for genetic analysis, and the questionnaire is completed, and the questionnaire is the same between both sites. So what next? There are 2 other clinical studies described for the iToBoS application. So we will do a second study with the Vectra, and the aim of this study will be to validate the AI tools that are being developed with the data we're collecting now. So that will be done again with UQ and Barcelona. And at UQ, we again will opt for a method where we piggyback on another study and contact research participants that are already having their total body photography, and seeing if they're okay with also consenting for their data to be used for this additional study as well. And in Barcelona, we'll do another prospective study because, as I said, the patients are coming in daily. So, and then the last study will be when we have the iToBoS total body scanner, and this will be placed at both Barcelona Hospital Clinic and also in Trieste in Italy. And so the objective there will be to recruit 75 patients each. And what we will need to do is collect the same data to create a new training data set so we can adapt the AI tools to the iToBoS scanner. And then as a secondary aim, we'll also compare the image quality and clinical usability between the Vectra and the iToBoS, so we can report on that and see where it needs developing from there. So that's all I have to talk about the research study. I'm very keen to hear any questions and also feedback, because that's useful for me as well when considering the next studies coming up. If it's negative, positive, or good, bring it on. So yeah, I'm done there.
Thank you for that, Claire. So who has questions? I always have questions.
I have loads of questions. I'll have a go at one initially. I'm just interested in the utilization afterwards, because as patients, we always are. I'm kind of, you've chosen high-risk patients. High-risk patients are going to be coming back regularly to be scanned, to keep an eye on things, because they always have this terror of a second primary, and they'd have the risk and all the rest of it. I'm wondering if, you're going to have a lot of patients who may be on immunotherapies and things. They're going to have things like vitiligo. I'm wondering if that's going to be something that could be quite useful, that this scanner can also pick up these kind of traits. We also know that there's some evidence that people who have vitiligo before they have melanoma may have better outcomes, all of those kind of things. And I'm wondering if the whole vitiligo thing, because I was just looking at some of your images there, and it would be quite cool for that.
I haven't considered the vitiligo before, so that's really interesting. Regarding patients on immunotherapy, that is one of the things we do ask in the questionnaire, is like, are you on any significant medication? And then we put in brackets, for example, immunotherapy, because that can certainly change things with regards to your skin changing quicker, basically, like melanomas and squamous cell carcinomas. So we do collect that information because we want to be able to more so for the otherwise it would be a bit of an outlier, if you know what I mean. Like it might skew the AI system, so we need the AI to go, okay, so that they might be different, like, because we're I just realized I forgot to mention, we do look at subsequent images as well, because we also want to train the AI to detect changes between 2 imaging sessions, whether it's 6 months apart or 12 months apart. So yeah, we want to also be able to let the algorithm know that this person is slightly different because they are having immunotherapies at the moment. So yeah, so we do collect that information, but I mean, we're kind of at the tip of the iceberg, like in that we're collecting this information and still not yet knowing its potential in a way, especially with genetic information. We're starting to understand phenotypes associated with specific genotypes, especially like the familial melanoma mutations as well. So the more kind of phenotype imaging information we collect, and looking at nevi distribution as well, like there's different patterns associated with different genotypes. So I mean, it's kind of, we don't we kind of know the questions, but yeah, it's still like we're just getting this data. And I think that a lot of that information on phenotype and being able to, know how we can use that is going to come once we have it. And I think one of the beauties about AI predictive algorithms, it can be data driven first. So a lot of the time, like depending on what methods you're using, but you don't need a prior hypothesis. You can be able to look at this data and then let the data tell you what is predictive of the outcome. So yeah, I think there will be things like that in the future. Probably not in this iToBoS project because we'll run out of time to do that. But yeah, maybe the iToBoS 2.
And worth thinking about the targeted therapies too, because I mean the changes in lesions on targeted therapies are quite significant too. So you'd want to be saying this is a change due to treatment rather than a change due to some progression or something. Yeah, really interesting. Thank you.
No worries. Thank you.
Any other hands or questions? Yeah, Violetta.
Yeah, well, good morning, everybody. Good morning, Claire. I was interested actually, and for our audience, how you define the high-risk population, what kind of patients are you going to enroll in these clinical trials, and what are their features?
Yeah, I think, well, there's the ultra high risk, if we're considering familial melanoma and testing for those genes. It would be personal and family history. High risk, generally we define, in Australia, it can be different because we've got a much higher incidence, but melanoma before the age of 40, like 1 melanoma before the age of 40 or 2 before the age of 60 is usually the criteria. But then especially in Australia, you need to consider their past sun behavior, UV damage as well. So yeah, but that's the main kind of cutoff. If you've had a melanoma before the age of 40, that generally puts you in the high risk category.
Yeah, thank you. And the second question is about the endpoints of the clinical trials. Especially for the validation clinical trial, I was expecting more comparison between Vectra and the actual iToBoS, like in terms of performance of the all device. But then I saw that you have only the comparative of the accuracy of the images, if I remember very well. So is this changeable, or this will evolve during time? You look for other endpoints. As I know, this is predefined. So I don't know how much flexibility you will have there.
Absolutely. That will be fleshed out more. That's because the protocol is not written yet. And I don't have a device yet. I don't know what the device does. So until I can actually see this iToBoS scanner, it's very hard to write a protocol on how I'm going to, compare the two devices. So that's key. Once there's an actual iToBoS machine, and if I can get a sneak look at what it can do, then I can design a protocol to compare what we can compare. So for sure, that will definitely be fleshed out more.
Thank you so much.
Yeah,
that is clear. Thank you.
Yeah.
So building actually a little bit on what Violetta just said. So 2 points actually. So one, a concern we're having is that what we see, at least in our communities, is that you have the activated patients going to get their full body scans. So you will have those at high risk who kind of get invited and get more encouragement by the clinical teams, but otherwise you have people who take own initiative. We actually have patients self-funding to get Vectra whole body images. So that, in our opinion, introduces a bias into the data set. So how do you think about that? We are, of course, worried if we then use that data set to train AIs on it. How do you think about that question?
Absolutely. I mean, so because I haven't been in Barcelona for too long, I think in Barcelona you will have that issue because it is the people, it's opportunistic recruitment. So you're recruiting the people that are coming in because they need or want a skin check. At UQ, we have had some other methods. So one of the previous studies that I'm recruiting from to use the retrospective data, it was a population-based study. So we were able to get the electoral roll records, like through big application process to be able to randomly select people from different age groups and post them out an old-fashioned letter to invite them to the study. So that is one of the studies we're recruiting from. So it is a more population-based one as much as we can. I mean, there's still going to be a bit of a bias for people that are more interested. And then the second study that we'll be recruiting from for the next study with iToBoS, it does have different risk categories. It is still biased to people that are interested in this type of thing, but it was a study that it started because we got a rather large grant in Australia to put 10 or 15 vectors across the eastern seaboard of Australia. And so we've got this huge cohort of people, and it received a lot of media attention. We're on the news and all that kind of thing. So we got a lot of people kind of writing to us going, how can we be part of it? So we've got a website there for people to submit their interest. So, and yeah, we've had no problem recruiting for that study. So so we are definitely getting people of all different risk spectrums there. And at least the UQ cohorts, they're not recruited from the clinic. We do get, our high-risk people do come from referrals. So especially in Brisbane, all the dermatologists, they all know our principal investigator, the director there. He's quite a well-known dermatologist there, and they refer all their high-risk people to us as well if they're interested in being in research, or even just having the vector scan as part of a high-risk clinical visit rather than research visit. So yeah. But I do think, yeah, we at least do get more low-risk people, whereas the Barcelona clinic will definitely be more high risk than that, I think.
Maybe that will be a topic for us to take up in the year and a half that we have left on iToBoS, because that is a discussion we also had yesterday, right? How do we reach the non-reachable or hard to reach population?
Mm-hmm.
We can't just do research or care for people like us. So I think maybe that would be an interesting one to reflect on the learnings that you've had in Australia, and where you've been very, very successful, to see how this could apply to the European context, which is also quite diverse. So maybe that would be something for our list to take forward. And the next question I had is so something that we have noted, because of course there are lots of apps that you can download, and you can imagine that as patient community, you get approached by lots of startups who want us to share their apps, and everyone takes pictures. And then of course there's always AI, and it all sounds great and fancy and all of that. And now the technical and validation issues aside, something that worries us and that we have seen as an issue is what we call vertical integration of an early detection device into our healthcare process. Because once you detect a lesion that is not okay, something has to happen. And this is often where our healthcare systems get stuck. We have different systems that actively encourage not to remove suspicious lesions until they're really suspicious. I mean, there are places that have quotas, how many a GP should remove, because pathology is costly. So what have you done on this vertical integration? So what happens if you pick up something that is suspicious, and what's the next steps?
Well, I can talk for more outside at UQ. I know that it depends on the patient as well, because a lot of people, they want that peace of mind of just having it out. So, I mean, it's not just, there is an issue of overdiagnosis, and there's also issues with, even within pathology, so much disagreement on what's a melanoma and what's a dysplastic nevi. And actually one of the studies with our cohort in Australia using the vectors across Australia is there is a pathology look at this to try to come to just to see what this variability is, and how we can fix this. So, I mean, I can't really because I'm not personally a dermatologist, so I can't really speak on behalf of them. But it is an issue, but and it is better in sometimes, if the patient is okay with it, to watch and wait as well, because there is this suspicion that there is these small melanomas that could potentially stay small for decades and never actually affect a person. So it's kind of it's a new area, it really is, but it is definitely something that's a hot topic for dermatologists for sure. And yeah, and definitely it's a consideration of what the patient wants as well. So yeah, I think that's all I can really say, not being a dermatologist myself.
We're obviously working on this, well, actually, even in a different European project like called Melkaya, where we work with pathologists, and this is into like pediatric melanoma, so that looks even different. So where we, of course, worry to miss something in a child is even worse than missing it in an adult. So, and also because they're harder to diagnose. So we've been discussing this, and I'm sure you've also seen, you know, publications on social media where people do like a very short-term follow-up on a very early stage melanoma and say this is horrendous overdiagnosis. Well, we all know that melanoma can, you know, come back decades later. We have people in our forums who had a primary 30 years ago, and then all of a sudden have a metastasis. So there is this time lag, and that's why we think it's a very important place for discussion. It's like no one has the answers, but that's why I think we think this needs airing and that needs a stage, and we all have to think about it, because of course overdiagnosis also does harm. So it's just like this, I think we think is a very, maybe that's another topic to take forward for all of us to think about, you know, how do we detect, what do we do next, how do we avoid overtreatment, overdiagnosis while not missing while we're doing this in the first place.
Well, I think that the integration of total body photography, capturing everything regularly, and the assistance of AI to be able to look at those changes over time, I think that is part of trying to address the issue of overdiagnosis and what's safe to leave, what should be taken out. So I mean, it's definitely an issue that's known, and yeah, so it's a hot topic for sure.
Yeah, with a condition to be affordable. It's
that's true,
and implementable, because probably will have, if it is successful, and other kind of tools, comparative tools with iToBoS will be successful and implemented, we will have a lag phase when everybody will get used and they will learn how to use it in the real practice and so on. But then will be a waiting time of several years or maybe more until you see fully implemented in the routine, in clinical practice on a routine base. And now I wanted to ask you something else. I know that you are not a dermatologist, but maybe I'm very curious if in Australia there is a reimbursement of the genetic test for people who are initially surveyed on or so on.
Yes. So there is, if you go through specific avenues, basically is the answer. So there's genetic services. So usually if you meet the criteria, you would need to be referred to genetic services, and then they can do the testing. And in Australia, they usually only would look at CDKN2A mutation. I'm not sure how much you know about the gene. So that's the main contributor. But I mean, with the affordability of panel testings, I think it's hard to justify only testing that one gene. If there's a history of ocular melanoma, then they check that one gene. So there is currently nothing to reimburse for a whole melanoma panel. I think, not that it's a good thing, but it will probably start becoming a thing in private dermatology clinics first, where patients that have the means can pay for that panel testing. You know, I think that's how it will start. The problem is such the high incidence rate in Australia that so many people would qualify for this type of genetic testing, and the shortage of the genetic workforce to be able to meet that demand as well. So like, I can't see them changing that from what it is at the moment anytime soon. And I think the wait time as well, because there is, you know, they prioritize to a point as well. So I don't think the melanoma testing, because it's not directly actionable, like it's informative, and it's, you know, it's from my research, it's good for the person to know generally, like and also it's good to identify a mutation within a family, so you can make sure other family members are aware as well. But yeah, I think just the combination of the high incidence rate, the genetic shortage of the workforce there, and yeah. So
what you meant that afterwards people will not be referred to a dermatologist for a scheme of follow-up or something like this. If they are identified with high risk, genetic high risk, they will not be referred to the dermatologist to be like, okay, follow up more regularly, or?
it's pretty still uncommon, genetic testing
Okay.
for familial melanoma in Australia. I can tell you in Brisbane, because the Brisbane dermatologists, they all know we're doing this type of research. They refer them to us, and then we can do it through research at no cost. But I don't think it's very common to even for dermatologists to refer their patients to genetic services. I actually, my research group, we did a survey of Australian dermatologists on their views and opinions on genetic testing, and they're just not too knowledgeable about, especially with melanoma, other diseases that are, as I said, direct actionable kind of results they're more aware of. But yeah, there is a paper published about it as well. So, but it was quite interesting. Like, I think it was only about 45% saw genetic testing as relevant to their position in the clinic today, but I think it was 86% saw it as relevant to their future practice. So I think everyone kind of accepts that it's a thing that's coming in, and a lot of our qualitative comments and feedback from that survey was, I'm interested in upskilling, but there isn't that, it's not there. So I think it is a gap that needs to be addressed, the upskilling and for dermatologists to know that this is available and how to provide it if they're
just before we have some more time for discussion now, but just for everyone, if you could look into the chat, please. We have a quick feedback form, because it sees this is now going on to a very different topic. We started with a clinical trial. This is already now basically prior to any type of scanner or clinical trial to it. So we would like to hear from you whether we would like to have some feedback on this session from you, and especially which topics you would like to hear more about. So the link is in the chat, so please use it. It's very short, I promise. It's no email or anything. Please use the form. You can submit more than once, but we would like to capture what you're interested in, where you think there are, I'd love to hear more about, because we are running out of time. But of course, we would like to continue this discussion. So please use the opportunity. And now, do we have any questions from anyone else we haven't seen yet? Good morning, everyone. You can turn on your cameras, I'm sure you have questions for Claire. That was quite a ride.
While everybody's thinking, I have one more question on the informed consent, kind of online informed consent, and the questionnaire online, because this is quite important. I'm quite surprised that there was quite a difference between what you could do in Queensland and what you could do in Barcelona, because I mean we see at places like the Champalimau Foundation in Portugal, where everything is on an iPad. And so there's these differences, and I think where is the time, because having filled in paper forms forever and ever for my own situation, I'm interested in knowing how we push this. Is there a carrot and stick kind of situation where you can get people to do these things online, or is it just going to be a block if you don't give them paper forms? And I'm wondering whether it's the patients or whether it's the system and the capacity of the clinicians that's the issue.
Exactly, because one of the comments I got was, oh, this won't go past ethics, so it's like, but what about the patients? What do they prefer? So I don't know, maybe the answer is we need to actually build the evidence. So perhaps a sub-study might be with our UQ cohort, is to have a proper survey to get that feedback from them. So and then if we have like published evidence saying like patients prefer this over this, or at least they prefer to have the option, or maybe that will help in going forward to ethics to try to convince them otherwise. So, but I think, cultural differences as well. And at the hospital clinic, I think that there's definitely an older cohort that I think they're seeing as well. So probably not willing to use the technology as much. So, yeah.
It's just, we seem to have to do it for banking, regardless of whether we wanted to or not, and yet in these other circumstances we sort of get given a choice because ethics says we can. So it is kind of odd, but yeah, interesting.
Yeah.
Any more questions for Claire, please? This is your chance. And as well, please use the form. Otherwise, I would like to now close the webinar. Thank you very much to everyone for being here, and that quite early, and that on a Friday morning. A special thanks to you, Claire. Thanks. That was really great to hear about all the work that has happened. And I hope we can, repeat this with maybe just like go into more detail into some of the issues, because I think you have touched on some rather interesting ones. Like you said, the training of clinicians was really important, and that may be a way to scale. That the online informed consent we just touched upon, I think we all have had issues in the past with this topic. I think that needs more work. We're obviously very much interested in this image challenge that is part of iToBoS, because we want collective learning to improve things faster. Familial melanoma is a big worry for us, including the genetic testing that Violetta mentioned. I think the population-based approach that you took in Australia is fantastic, because that addresses some of the issues that we always have. So I think we would love to hear more. And then how do we integrate this and have like a holistic approach from genetic testing? How do we follow up these people? What do we do then if we find something that is worrisome? And how do we kind of treat them effectively? I think that's quite a number of things to take forward. So with that, thanks a lot.
Thanks for having me.
Thank you so much.